Good morning and welcome to our webinar today. My name is Ryan Budd. I'm the Director of Partnerships and Events with Life Sciences BC. Happy New Year to everyone. Just to let you know, all attendees have their video off and are on mute. However, we do encourage you to be very interactive and to post questions in the Q&A section, where you can also upvote on any of the questions. And we also have the chat function there. It is now my great honor to introduce Wendy Herbert, the President and CEO of Life Sciences BC, to start the event. Over to you, Wendy.
Thanks, Ryan. Hi, everyone. Happy New Year and welcome to Life Sciences BC's first event of 2022, presented by the Gairdner Foundation. Thank you all for joining us today. As we begin, I would like to acknowledge that I'm joining this event from within the ancestral, traditional, and unceded territory of the Coast Salish peoples, including the territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. I'd also like to acknowledge that others are joining us from across this beautiful province and other unceded Indigenous territories around the country. We're excited to be here today. I think I've been saying that now for 2 years. Over the past couple of years, Life Sciences BC and our community have fortunately embraced virtual events. We're grateful that we have the opportunity to continue to discuss and draw attention to the extraordinary work that's being done in the life sciences sector across BC and Canada. For those that do not know, Life Sciences BC is a not-for-profit society. Our members consist of academic and research institutions, small and large companies operating in the life sciences space in the areas of digital health, medtech, med devices and diagnostics, therapeutics, and other organizations that provide support and services to the broad life sciences ecosystem. Of course, very importantly, as part of that are the researchers, which is where it all begins. We're funded through membership and generous contributions of our sponsors and partners. This event was made possible by the support of our sponsor, the Gairdner Foundation. Thank you for your investment in Life Sciences BC and the life sciences ecosystem. It's invaluable and we're very grateful for your support. At each event, we like to introduce our newest members. Today, we would like to introduce EM Pharmaceuticals, Molecular Forecaster, and the UBC MRF program, which is based out of Okanagan. Welcome. We're always welcoming new members, so please reach out to any of us at LSBC or go to our website, lifesciencesbc.ca for more information. So we're excited about today's event. We're going to include a lecture on how gut peptides drive therapeutic innovation for individuals with metabolic disorders. We're going to start the program with me introducing Dr. Janet Rossant, who will introduce Dr. Daniel Drucker, our keynote speaker. Following Dr. Drucker's talk, Dr. Rossant will moderate a Q&A. So as Ryan mentioned, please add any questions that you have to the Q&A, as Dr. Rossant will be monitoring that throughout the talk. So now to introduce Dr. Rossant, and you both can turn on your cameras now. Dr. Rossant is a senior scientist and chief of research emeritus at the Hospital for Sick Children in Toronto and president and scientific director of the Gairdner Foundation. She's an internationally recognized developmental and stem cell biologist, exploring the biology of early embryo and its stem cells and their applications to understanding and treating human disease. She has also been actively involved in ethics and public policy discussions around stem cell research and genetic modifications. She led the Research Institute at the Hospital for Sick Children from 2005 to 2015. She's received many honors and recognition for her extraordinary work, including 6 honorary degrees, an election to the Royal Societies of London and Canada, and the US National Academy of Sciences. In 2018, she received the North American L'Oreal UNESCO Women in Science Award. So a very extraordinary, accomplished leader. And please join me in welcoming Dr. Rossant.
Thank you very much, Wendy, for the very kind introduction. And of course, I'm really delighted again to be here on behalf of the Gairdner Foundation and partnering with LSBC on this event. We've had an ongoing partnership now for several years, and I'm really pleased to be here. Just for those of you who are not so familiar with what the Gairdner Foundation is, I just want to give you a short background. So Gairdner Foundation, really, we celebrate and recognize scientific excellence in research that impacts human health. We do that through the annual Canada Gairdner Awards. And these awards go to the very best scientists around the world whose research has really changed the way we look at the development of human health and the impact on our future health of society. More than 400 awards have been presented around the globe. 97 of those have gone on to receive the Nobel Prize, including just this last year in October, Dr. David Julius, who was one of our Gairdner awardees. So, but we don't just recognize excellence, we like to build on that to bring to Canada the world's best scientists to present their work across the country in events like this one. And we also seek to inspire the next generation of young scientists and innovators. So today you're going to hear in this Gairdner presentation, you're going to hear from Daniel Drucker. And Daniel is one of the 2021 Canada Gairdner International Awardees. And he was awarded, along with Dr. Jens Juul Holst and Dr. Joel Habener, for research on glucagon-like peptides that has led to major advances in the treatment of type 2 diabetes, obesity, and intestinal disorders. And you'll hear much more about that in his talk. Dr. Drucker is a professor in the Department of Medicine at the University of Toronto and a senior scientist at the Lunenfeld-Tannenbaum Research Institute in Sinai Health. And that's where he's speaking to us today. His lecture is called Gut Peptides Drive Therapeutic Innovation. And please, if you have questions, just pop them in the chat or the Q&A, and we'll have time to talk to Dr. Drucker after the lecture. So Daniel, the stage is yours.
Thanks very much to Gairdner and to Life Sciences BC for the opportunity to share some thoughts with you today. It's I'm sure it's nicer in BC than it is in Toronto today. It's a balmy, minus 20 below in Toronto. So enjoy your weather there. And these are my disclosures. We've interacted with a lot of companies over the years in regard to translation and drug development. And they've actually been very useful in helping us move things from the bedside to the bench and back in terms of interrogating mechanisms. So how did this all get started and what's the story about? It really all got started with this gene that you see at the top in yellow, the glucagon gene. And this is really a summary slide of the journey over the last 35 years, but understanding how these peptide hormones work has led to the development of GLP-1 receptor agonists for the treatment of type 2 diabetes and people with obesity, DP4 inhibitors, very widely selling tablets for the treatment of type 2 diabetes, and GLP-1 receptor agonists for the treatment of short bowel syndrome. And we'll discuss, how we got there and where we're going. If you have to leave and you don't want to miss out on the future, here's a summary slide of, I think, what the next developments will be in terms of therapeutic innovation in the clinic. I think we have trials underway now for Alzheimer's disease, for non-alcoholic steatosis. We have new combinations of these GLP-1 drugs that are clearly more powerful than GLP-1 alone. And I think we're going to see many of these introduced in the next few years because the phase 2 data looks very promising, and even some of the phase 3 data has been reported. So we're not, I think, at the end of the story. We're probably a third or halfway through in terms of how these peptides will change the landscape. So how did I get into this? I wish I had been clever enough to choose to work on the glucagon gene. I was just fortunate enough to choose the lab that was working on this gene in the early 1980s. Some of you know I was going to this lab in Boston to work on thyroid hormone. And when I got there, I was told that was not an option anymore. And I was assigned to work on the glucagon gene. And we didn't know anything about these glucagon-like peptides. They were just sequences sitting there in the gene with unknown function. And one of my first projects was to say, well, are they actually made? Can somewhere in the body some cells or tissues liberate these glucagon-like peptides if we express them as cDNAs, for example, in different cell lines? And here's some of the work that I actually started to do in 1984. And when I looked at the post-translational processing, it turned out there were multiple forms of GLP-1. And that turned out to be very important because the shorter forms, such as GLP-1 7-37 or 7-36 amide, those are the bioactive forms. But the conventional longer forms, such as 1 to 37 or 1 to 36 amide, they don't interact with the GLP-1 receptor really at all. And that sort of sidetracked some people for quite a while in terms of figuring out what these did. Now, one of the other very simple experiments that I was fortunate enough to do about 1985 was simply to try and understand what GLP-1 did. And I used a whole bunch of cell lines that were in the lab, including islet cells. And here's the islet cell data. And we can see that the short form of GLP-1 increases cyclic AMP accumulation and insulin gene expression and insulin secretion. And it does so in a glucose-dependent mechanism of action. And to be honest, I did not have the vision by myself to know that decades later this would be therapeutically useful, but I must give Joel Habener, one of this year's Gairdner awardees with me, and Professor Holst, who really did have the vision at this time. Now, one would think that with all of the world's brilliant biochemists, that we would have these amazing designer forms of GLP-1 that would be used for the treatment of type 2 diabetes. And the answer to that thought is yes, we do. And the chemists have been brilliant, but they weren't first. In fact, the first GLP-1 receptor agonist that was approved came from this lizard, Heloderma suspectum, also known as a Gila monster. And it's a naturally occurring protein isolated by John Eng at the Bronx VA in the 1990s. This is a poisonous lizard. So for those of you thinking of a gift for Valentine's Day for your cherished partner, it probably shouldn't be this lizard, because if this lizard bites you, it's lethal within minutes. But among the peptides in its venom is this GLP-1-like protein. And through a whole bunch of circumstances that are kind of amazing, this became the first GLP-1 receptor agonist approved worldwide in April 28th, 2005 for the treatment of type 2 diabetes. And I don't do clinical investigation very often. I'm really more of a lab person, but it was intriguing to me the possibility that one could develop this peptide technology in a longer acting form, because there was no once weekly drug ever for the treatment of type 2 diabetes. And so I actually led the first phase 3 trial to test the actions of once weekly exenatide, and it worked. It was quite remarkable. It was very effective. And now we have multiple once weekly GLP-1 receptor agonists available for the treatment of type 2 diabetes. And some of them are shown here, and they've gotten better and better over time. We've optimized as a community the pharmacokinetic and pharmacodynamic properties. And as a result, you can start to see hemoglobin A1C reductions, a measure of how glucose is lowered in people with type 2 diabetes, that really are the same as what we achieve with insulin in head-to-head studies. Of course, the major point of differentiation with these GLP-1 drugs is shown here, and that's the weight loss. With insulin or sulfonylureas or thiazolidinediones, we really don't see weight loss. In fact, we sometimes gain a bit of weight. But we're now starting to see 5, 6, 7% mean weight loss with these drugs when used for type 2 diabetes. And additional innovation in this space has allowed for the development of an oral GLP-1 drug. Quite remarkable, because we know that the gut, the stomach, is designed to break down peptides. That's the process of digestion. When we eat, we have enzymes, we have a low pH, we have a mucus layer, we have microbiota with their byproducts that can degrade proteins. We have tight junctions to prevent these small peptides from crossing the epithelium. Yet remarkably, if one uses an absorption enhancer developed by Hemisphere Biosciences in the United States and couples it with semaglutide, we can get very efficient absorption of this combination. And this is now approved as an oral once daily therapy for diabetes. And we can see, if we compare the circulating levels achieved with the oral form in the Pioneer program versus the circulating levels achieved with the injectable form in the Sustain program, that we see identical pharmacodynamic relationships with oral versus injectable drugs. So as long as you get the drug in there, it doesn't really matter how you deliver the medication. Now, a huge chapter in the story of the GLP-1 receptor agonist has been the safety results we've seen in the outcome trials. And to summarize, these drugs reduce heart attacks, strokes, cardiovascular death, and all-cause mortality. And this is pretty consistent across the trials and extremely reassuring, because this is really the major reason why we treat type 2 diabetes. It's not to lower the glucose per se, but it's to reduce the development of these complications, which in fact is a property of this class of medications, as well, we now know, of the SGLT2 inhibitors. So how does this all work? And this is something that still puzzles me in our lab. We've been able to use the genetic technology, in fact, pioneered by Dr. Rawson, our chair of the Gairdner Foundation, decades ago, to try and tease out where the receptors are and inactivate the receptors and do some pharmacology and physiology. And we're making slow progress, but it's still a puzzle. So it's widely thought that GLP-1 might reduce atherosclerosis as a contributing mechanism to those reduction in rates of heart attacks and strokes and heart and myocardial disease that I showed you. But where are the GLP-1 receptors that allow the GLP-1 receptor to reduce atherosclerosis? In animals, if I give ApoE knockouts or LDL receptor knockouts, or animals treated with a PCSK9 adenovirus, if I give all those animals that develop atherosclerosis a GLP-1 receptor agonist, I get beautiful reduction of atherosclerosis. But it's been difficult for us to tease out exactly the precise cellular sites that contain the GLP-1 receptor that are important for attenuating development of atherosclerosis. So we've taken out the GLP-1 receptor from immune cell populations, from endothelial cells. We still see reduction of atherosclerosis when we give the GLP-1 receptor agonist. How does GLP-1 reduce circulating triglycerides, which it does very powerfully? It actually flattens in humans chylomicron secretion when we eat a meal. And there are no GLP-1 receptors on enterocytes. So how does this work? Is there some sort of neural communication that conveys a GLP-1 signal from a neuron to an enterocyte? And we've taken out GLP-1 receptors from virtually all of the neuronal populations in the central nervous system, the enteric nervous system, the autonomic nervous system. And when we do these studies and give olive oil to mice, and with or without multiple GLP-1 receptor agonists, we still reduce chylomicron secretion and triglyceride levels. So what I'm trying to convey is that I know what GLP-1 does in humans and in animals, but it's still a real challenge for me to mechanistically identify the precise pathways through which it exerts its action. Now, GLP-1 also reduces inflammation in animals and in humans. And again, how does this work? Here I'm showing you macrophages from multiple different tissues with and without induction of inflammation. I don't see the GLP-1 receptor. Where is the GLP-1 receptor in the immune system? It's actually very robustly expressed in a population of gut lymphocytes we call the intestinal intraepithelial lymphocytes. And this is a big focus of study in our lab. And this makes a lot of sense because the gut, particularly the distal gut and the colon, is obviously a huge site of inflammation. We have viruses, we have fungi, we have the gut microbiota. Many of their metabolites and cell wall products actually promote GLP-1 secretion, which in turn talks to these receptors on these local lymphocytes to modulate inflammation. So that makes a lot of sense for gut biology. But we know that GLP-1 also reduces inflammation in many distal sites that are not characterized by abundant GLP-1 receptors, where we do not find infiltrating immune cells that express the GLP-1 receptor. So there must be inter-organ communication that conveys this anti-inflammatory response. Very exciting stuff that we're still making progress on today. So what are we going to see next for GLP-1 therapeutics? Well, we might just see approval for NASH, people with non-alcoholic steatosis, often metabolic liver disease associated with obesity and diabetes. Here's some phase 2 data with Semaglutide. demonstrating reasonable reduction of NASH as quantified on histological scores, and some attenuation of the progress of fibrosis. So how does this work? Most of this is probably indirect. It's probably due to weight loss. But if we knock out the GLP-1 receptors on a population of immune cells, and then look at the benefit we see on attenuation of liver inflammation, we see a reduction in the benefit of the GLP-1 receptor agonist. And so there's less fibrosis, there's less reduction of liver fat, there's less reduction of inflammation. And so I've tried to now reorient my thinking into saying that weight loss is probably the primary driver of how GLP-1 benefits NASH, but there may be an additional anti-inflammatory component that's weight loss independent. There's also been tremendous progress in the use of GLP-1 as an agent to treat people living with obesity. And this follows the biology that I've summarized here, that there are multiple distributed GLP-1 receptor populations in many different regions of the brain that are capable of transducing a signal that reduces food intake and ultimately promotes weight loss. And this has resulted in 2 different GLP-1 receptor agonists so far being approved for the treatment of people with obesity. And we hope to see sustained benefits as shown here on the right in these outcomes. And there's actually a large outcome study underway, the SELECT trial, using semaglutide 2.4 milligrams once a week to see if we can actually reduce heart attacks and strokes and cardiovascular death. This is the phase 3 data for using semaglutide once weekly to treat people with obesity. You can see now that the weight loss achieved is in the range of 15 to 18%. We're not quite at bariatric surgery levels of weight loss yet, but I think we're making real progress in offering real meaningful options to people who don't choose to have bariatric surgery. And this is now approved in the United States, as well as very recently approved in Canada for use in people with obesity. There's also great enthusiasm for combining other agents with GLP-1 to make what we call co-agonists or triagonists that can activate multiple receptors and potentially bring even greater benefits to the clinic. And the drug that will come next, which has been extensively studied in phase 3 and is now before the Food and Drug Administration, is tirzepatide, developed by Eli Lilly. It's a GIP-GLP-1 receptor co-agonist. It's even more powerful than the GLP-1 receptor agonist alone. And we are now seeing weight loss with this GIP-GLP-1 receptor co-agonist that's double digits in people with type 2 diabetes, and probably will easily approach 20% or more in people with obesity. And those trials now are underway. And this drug will probably be approved in the United States in the summer of this year, and hopefully not much later in Canada. Lots of attention to other coagonists. Here I'm showing you the results of an amylin receptor, GLP-1 receptor coagonist, optimizing amylin to be an acylated, degradation-resistant, once weekly administered molecule called Cagrilintide, combined with once weekly semaglutide. And again, this is projected. You can see the lines for the extent of weight loss are still going down quite steeply at 20 weeks. And so most of us think this is easily going to achieve 20 to 25% body weight loss, and it's also in phase 3 clinical trials. So let me turn my attention in the last few minutes to GLP-2. It's part of the same glucagon gene. It's related in sequence to GLP-1, and its role is to prepare the gut for nutrient absorption by enhancing nutrient absorption directly, as well as expanding the mucosal epithelium. And these are the very simple experiments that we did in the early 1990s to show that GLP-2 is a bowel growth factor. And after a lot of frustration and some disappointment, but occasional success, this is the results of the second phase 3 trial using a GLP-2 analog developed in our lab called Teduglutide. And you can see that this reduces the number of nights per week that people need to spend requiring intravenous nutrition. And many people can really almost come off parenteral nutrition or markedly reduce their time with this intravenous overnight feeding. In the real world, about a quarter of people can completely come off parenteral nutrition. And nothing is like N of 1 results, but this young woman who was part of the pediatric trial in Boston, and Teduglutide is now approved as well for pediatric short bowel syndrome. Her story, Ava's story, is now part of the Boston Children's Hospital website, and it completely transformed her life from a young girl that needed to be hooked up to intravenous feeding every single night of the week, and now she doesn't need it at all, and she has a fairly normal life. So this has been a lot of fun with the basic science starting in the early 1980s and continuing to this day with many milestones and really hundreds and thousands of colleagues contributing to this story, both at the level of the basic science, the modulation of the molecules for pharmaceutical development, the clinical trials, and of course, the tens of thousands of people that have volunteered for the clinical trials. They have all contributed to this story, and my thanks go out to all of them, as well as my colleagues Jens Holst and Joel Habener, who made extremely important contributions along the way. So delighted to share a small bit of this story with you. I think you can perhaps share my excitement, not only for what has been the story to date, but what is coming next, I think might be even more valuable for people living with metabolic disease, and be delighted to end here. Thank the people in my lab who are doing the experiments today, and to take some questions for anyone who wants to quiz and share some of the excitement I have with questions, etc. Over to you, Dr. Ross.
Thank you. That's a great talk and a great story from the sort of beginning of the science right through to the current clinical implications and the clinical trials. So I'm opening up the floor to questions from the audience. Please put them in the chat or in the Q&A, and I will pick them up there and pass them on. Meantime, I'm going to start with a couple of questions of my own. I guess every time I hear you speak, it seems even more exciting that the future therapeutic applications for these peptides is just growing all the time. And you talked about some of them, and you mentioned some that would be of even more potential impact, but didn't dig deep. So talk about Alzheimer's. So what's the story there, and what is the potential in Alzheimer's?
Yeah, so probably about 20 years ago, a neuroscientist, Matt During, called me up, and a lot of G protein coupled receptors, as you know, by signaling through cyclic AMP and other pathways, are neuroprotective. This is not unique to GLP-1, and he wanted to do some studies, and we gave him the GLP-1 receptor knockout mice that you helped us make a few decades ago. And if you gave GLP-1 to these mice, they had smaller strokes and better learning and memory. And if you did the same experiments in the knockout mice, they had bigger strokes, a larger hemorrhage in the brain, and poor cognition. And again, I didn't think much of that, because that's the story that goes on in preclinical stuff. But if you started to look at the clinical trial data, there were fewer and fewer people who would be reported with the diagnosis of cognitive dysfunction or Alzheimer's disease in the trials in the arms that received GLP-1. And we have now about 100 or 200 papers looking at the neuroprotective biology of GLP-1 in all of the various Alzheimer's models that people use, which we know are not predictive for Alzheimer's therapeutics. That's an extremely difficult area to work in. As you know, the Gairdner has really invested a lot of time and energy in this. But I think Novo Nordisk has basically rolled the dice and spent a lot of money, and there are now 2 phase 3 trials underway with oral Semaglutide in people with cognitive dysfunction at high risk for the development of Alzheimer's. And we're very fortunate that the biomarker field has really come a long way. And so we can now enrich these trials for people at very high risk of progressing from cognitive dysfunction to Alzheimer's. This will probably take 3 to 5 years. If you were a betting person, you would probably say that there's a 95% chance that this will not work, because that's been the story to date. But the only way that we really test these theories is by doing these trials. And so it's a high risk strategy, but I'm delighted to see that Novo Nordisk is making the investment.
Well, you could envisage that even without having a sort of direct neuroprotective effect, that just the effect of being able to reduce diabetes, get weight loss, reduce the overall risk of strokes and other vascular events, because obviously a lot of dementia is caused by small vascular events. So those could be reduced without having a sort of specific major effect that's neuroprotective.
Yeah, that's 100% correct. So if you didn't know about senilin or tau or amyloid or any of the targets, and you just could reduce brain insulin resistance, improve connectivity, reduce brain inflammation, improve glucose metabolism in the brain, all of the things that GLP-1 has been shown to do, you could potentially make an impact. And I think that's part of the story, certainly.
Yeah. Okay, so you talked, I'm waiting for the audience. Come on, audience, give me some questions here. Dan and I talk quite a lot anyway, but we can have another discussion. You talked, and particularly in this talk, about the new oral forms of the GLP-1 agonists. How important do you think that is to really make the use of these products more widely available?
Yeah, that's it's an open commercial question. So to date, these drugs have been injectable and they've been the domain predominantly of the specialist, the endocrinologist. But 95% of people with type 2 diabetes worldwide are treated by their primary care physicians because they don't have access to endocrinologists, and diabetes is so common now. It's really a very large proportion of primary care. And primary care, I think, has more comfort with oral medications for the treatment of many diseases. They're not quite as experienced with injectables. Some primary care doctors are, they're fantastic with injectables, but I think the ability to have an option with an oral therapy, and we're starting with oral semaglutide, which is a peptide, but there are several oral small molecule GLP-1 receptor agonists in phase 2. And these will be, I think, very useful because the cost will be much lower and it will be, I think, much more affordable for many healthcare systems. So I think it's an open question how successful the oral forms will be. It will take a few years to answer that, but I think ultimately they'll probably be more popular than the injectables. It just might take us 5 or 10 years for that to transition.
Mm-hmm. Okay, I do have a couple of questions coming in on the chat and the Q&A. One from Sanjeev, who asks whether the same process, I guess the same action, could be used to help with spinal cord injuries.
So yeah, there is preclinical data in this exact realm of spinal cord injury, both sort of mechanical spinal cord injury or experimental myelitis and transverse myelitis, et cetera. And GLP-1 does work. I'm always very reluctant to put too much emphasis in the animal data. That seems paradoxical because that's all I do is animal experiments. But we know that it's so difficult to go from much more simple reductionist animal models of disease to the greater complexity of human disease. So I don't think we have any human clinical data that I've seen in people with spinal cord injury. And it would be fascinating if somebody wanted to take that on. I think it would be difficult, but one doesn't know until one does pilot studies.
Okay, a sort of a similar kind of question from Delia Cooper, who asked whether GLP-1 has any possible effect on patients with RA.
Yeah, so it is anti-inflammatory, and I actually tried to do a randomized controlled trial in people with psoriatic arthritis and psoriasis for many reasons. And I teamed up with Professor Daphne Gladman here in Toronto, who's a world expert on psoriatic arthritis. And we got the trial funded. And it sort of coincided about 5 years ago with this wave of new biologics that we all know have been amazing in reducing disease activity and almost extinguishing for many people joint inflammation in people with rheumatoid disease or psoriasis, whether they're TNFs or JAK inhibitors or multiple different molecules. And so we couldn't really enroll. So the answer is, I don't really know. We haven't seen compelling clinical trial data. Again, weight loss alone or anti-inflammatory properties alone might contribute to improvement, and there are case reports, but we just don't have really good RCT data.
Okay, and from Jonathan, which I think it's an interesting question, more general one. What things can be done to create more awareness of these treatments beyond primary care docs? Is it the role of associations, diabetes, Alzheimer's, or public health? I mean, how do we make the awareness of these treatments more broadly?
I think, yeah,
ads on TV, I have to say. So I mean, companies obviously are also doing some.
Yeah, if you watch professional sports or the financial news or whatever, I don't know what it is that I watch, but I don't watch much, but I see the ads on television. I think that the awareness is really starting to increase now because of the new results we see for weight loss. And so, in fact, it's almost remarkable that Semaglutide once weekly, which has a trade name mysteriously known as Wegovy. There's somebody who thinks that's clever, but it's having manufacturing problems, and the company can't meet the demand for this weight loss drug. And I think this has really raised the attention. We've seen now lots of articles in the New York Times and the Wall Street Journal and the popular media, the Financial Times, and the international press, because you're starting to get, in these weight loss trials, you're getting 20% weight loss in 40% of the people. And that's unprecedented for medical weight loss therapy. And I think we're going to do even better and better over the next few years. And given the challenge we've had with obesity, I'll tell you an anecdote. I had somebody from the RAND Corporation, which is sort of one of these industrial think tanks that has a lot to do with the US military and global security. And this person asked me to write an article with him about how the use of these weight loss drugs would improve the military capability of the US military, because they have a problem with overweight and obesity. And I said, thanks very much. I don't think that's really what I'm interested in doing for multiple reasons, but it shows you that the weight loss is catching people's attention. And as we get the new data for NASH, and let's see what the data shows for heart failure and for Alzheimer's disease, I think the awareness will gradually increase. But the drugs are still new, they're still expensive, there's still barriers to use. And so it does take time until they become more widely used.
And just because we're in Canada, can you give us a feel about the sort of relative usage of these drugs in Canada versus the US?
So they're actually very popular in Canada. I think, we know this week, I might as well wear my Insulin 100 hat. So this week is the 100th anniversary of the first administration of insulin to Leonard Thompson at Toronto General Hospital. And, Canadians have always, I think, been very quick to both innovate in diabetes and to adopt innovation. And GLP-1 does very well in Canada, both from an investigational point of view, as well as from a use point of view. So these drugs have been very popular. The oral form of Semaglutide has been used in Canada for almost 2 years now, and other countries look to us to learn about adoption. So I think it's been very popular in Canada. There were reimbursement issues. We have, as I'm sure all of the members of this association know, we have a lovely agency in Canada that's called CADTH, which is the Canadian Association for Drug Technology and Health Assessment. And CADTH was originally quite reluctant to recommend reimbursement of these drugs because they were expensive. But I think over time, there's been some compromise. Once one looks at the benefits that are in a sense unparalleled, there's been a gradual introduction into many of the healthcare programs and reimbursement much more widely now. But for the first few years, I would go to Ottawa and I would give the same sort of spiel about, look how amazing this innovation is. And they would just kind of stare at me with frowns on their faces, and the reimbursement recommendations were not very favorable at the beginning. So it takes time, I think, to knock down these doors, and it takes a lot of evidence as well.
Great. So the next question, which is more of a more scientific one, really. There's a trend to pinpoint which time of day is best for the effectiveness of medications. Given what's known about the biology of GLP-1, is there an expectation of circadian influence?
Yeah, so great question. And, there's every week there's a new app or a new book on the circadian code and when you should eat what, et cetera. So very popular. And the L cell that makes GLP-1 actually has its own autonomous circadian pacemakers. And so many of the same clock genes that you're familiar with in the hypothalamus or in the liver are also autonomous in the L cell. And GLP-1 secretion is regulated by the clock in humans. And this work has really been done by my colleague, Patricia Brubaker, a professor in physiology at the University of Toronto. What's less clear is whether the GLP-1 receptor is differentially responsive in the context of circadian rhythms. And so we have much less data to support that possibility. And remember, these drugs now are given so that they're available continuously 24 hours a day. So we've really removed the rhythmicity from the pharmacology. And so the question is, could we somehow capture differential biology if we were to use these drugs at different times of day, different, morning or evening, or in relation to meals, not meals? We don't really know, but that's not how they're used pharmacologically now. But it's a great question to answer scientifically.
Mm-hmm. Great. So I think that's the end of the sort of specific questions. So I'm going to turn, since this is about life sciences and innovation, I'm going to ask you some general questions about your career, your progress as an innovator. So you're a basic scientist, you're a clinician, you mix those two together, but you've also obviously been pushing and making sure and working with people to ensure the sort of commercialization of these products, because that's the way to get them into people. So can you give a little bit more about your relationships with companies? How often did you find yourself being the person who's driving the commercial interests, or how often do you just hand it off, but you have to hand it off to the right people? So, and this is sort of a general, what would you tell other people to do?
Yeah, so there are multiple ways to cross the finish line. But I think what's very clear, if you want to cross the finish line, and the finish line, I'm using the metaphor as an approved product that reaches the marketplace. Whatever that technology is, whether it's drugs that have to go through a regulatory process or it's products that have to be cleared for health and safety and efficacy, it needs a champion. It's really difficult to hand off any technology or any potential future product to an organization where there is not one champion or one or two champions driving the development of that technology. Because without a champion, there are often competing technologies, things that are also attractive. A company has often got 6 or 10 molecules that it could develop, or 6 or 10 potential products, all of which are competing for investment. And the investment is so prohibitive often that not all 6 or 10 can be resourced. And so unless you have that champion that's always beating the drums for the merits and virtues of this particular technology, it is often very difficult to cross the finish line. And so clearly I've been the champion for the molecules that I've been involved with, but I've generally been an external champion. And it's been very clear to me that unless there were internal champions in the companies that I've licensed these products to, it's often much more difficult. So there has to be this constant sort of person who's the mother or father or parent of the technology as it moves through the various stages of technology, because there will be hostility, there will be competition, there will be people in the room who would rather their vision or their technology be developed versus the one that you've put forth. And so it's not always easy, and sometimes things get put on the shelf for 2 or 3 years. I think the GLP-2 story is really a good example. So it was licensed to a company that was founded by John Evans, one of the great pioneers of Canadian science. and it was called Allelix. And when Allelix licensed in GLP-2, they also had PTH for osteoporosis. Osteoporosis is a huge multi-billion-dollar market. PTH got all their love and attention. GLP-2 really got put on the shelf, to be honest, and it wasn't long before PTH ran into some difficulties and we were banging the GLP-2 drum, and GLP-2 got back on track, et cetera. So there are many stories like this where you really need to have constant love, nurturing, attention, fostering the development of your technology, either externally or internally. Otherwise, there's a good chance it will hit a speed bump, it won't have a champion, it will get put on the shelf and may not make it.
That's great. I think that's a great stories, but it resonates obviously with lots of other people who are trying to take their products through to market. And just to finish then, maybe a more general question about the sort of the life science innovation environment in Canada. And what can we do better in Canada? We're doing well at the moment, I believe, but what can we do better? What are the roadblocks to Canadian innovation?
So I'm probably the least qualified person in the audience to answer this, because I know we have a lot of tremendous expertise in the various members of Life Sciences BC. But I'll just give my simple perspective. I think our basic science is amazing. I think really we have, whether it's at UBC or McGill or Toronto or dozens of Simon Fraser, so many universities, we have fantastic science. But I think what we don't have compared to, let's say, the Stanford area or the Boston area, San Francisco, San Diego, et cetera, we don't necessarily have the depth of incubators. We don't have the same depth of venture capital, and we don't have the same number by any stretch of pharmaceutical entities that have the expertise in taking these technologies to the market. And so it's gotten much better over the last 25 years. When I first started, the technology transfer office at Toronto General Hospital wasn't even an office. It was just part of the vice president of research, and it was Don Lane and Monique Evis, and I think there was a filing cabinet, and that was the technology transfer operation. Today we have large numbers of professionals who are excellent at what they do, and we have ecosystems and we have groups for mentoring, but we're still lacking the deep VC financing, and we still are lacking the second and third tier and major multinational large pharmaceutical companies. And that's a complex story. That's sort of taxation and federal policy and investment at multiple levels, and so many different things that factor into that. And that's probably a whole 1 or 2 day symposium by itself, with many more people more qualified than me to address.
Okay, well, I think that's a good way to end the discussion. And I would like to thank you very much, Daniel, for again another great story and another great discussion. And I hope that the audience has enjoyed hearing Dr. Drucker's story, and I think there are many parts of his story that resonate. And of course, it's a great success story, a great Canadian success story. So congratulations again.
Thank you.
Wendy?
Thank you very much. And I want to reiterate what you just said. It really is a great Canadian success story, and you referencing it back to the 100 years ago of insulin discovery. Canada has great deep roots in scientific research, specifically around this particular state. I really enjoyed not only the discussion on all of the exciting innovation and research that's happening. I spent a good part of my career at J&J in the diabetes space, so it's super exciting to get caught up about what's been happening with GLPs. But I also enjoyed the conversation that you brought it forward through to the innovation and actually bringing this innovation to patients, to patients where they can benefit from it. And some of those challenges in scaling innovation, I would agree with you. We've come a long way in tech transfer, but we also have a lot of opportunities ahead to accelerate the amazing world-class research that happens in Canada and trying to get it into patients for the benefit of really what it's all about, that we're all trying to create better outcomes and sustainability within our healthcare system. So thank you both for your leadership in this space. It's critically important, and we're so appreciative that you've taken the time to share that with us today. And so with that, thank you to the Gairdner Foundation. As Janet said, we've had a long history of collaboration, and we look forward to continuing with that. And Dr. Drucker, thank you for your fascinating lecture. We look forward to hearing more and following your research and innovation. At each event, we like to talk about what our next events are coming up. So I think Claire is about to project those. So next week, actually, we have our 3rd annual Career Connect Day presented by BioTalent Canada. This is to bring together the entire ecosystem to learn about careers. We've got profiled companies, people talking about their careers and what they've accomplished and how they've ended up. Some very interesting stories from some of our leaders. And BioTalent Canada will be presenting their latest labor market analysis, which is very interesting as we look ahead to the life sciences sector and the importance of talent development and training. On January 27th, we will have a McCarthy Spotlight speaking series. The theme will be around oncology. We have some of our leaders in British Columbia speaking about that. February is Access to Innovation, our annual conference with the theme Building on Excellence, a Vibrant Future for BC Life Sciences. That is where we will continue the conversation that Dr. Drucker was just having around scaling innovation and tech transfer. And in March, we have a new event that we're having, which is called Invest in the West, presented by Amplitude Ventures, where we will have a number of our SME companies from British Columbia and across the West presenting and showcasing their innovation to investors and to the community at large. So with that, I'd like to again thank everybody for coming. We're grateful for your support. We want to acknowledge our sponsors and partners. In particular, our Platinum, Admare, Ferris, GSK, Janssen, Pfizer, and Roche, and Gold, AbCellera, Blades, Innovative Medicines Canada, Novartis, and Stem Cell. Thank you all. And again, this was our first event for 2022. So Happy New Year to everybody. And we look forward to seeing you at future events.